Hello, everyone. Thank you for joining our webinar today. I'm excited to have Joelle here with me hosting the webinar. I am Amy, and I will be moderating today's session. Before we begin, I just wanted to let you know a little bit more about how the webinar will run. The webinar is being recorded, and the slides and recording will be available to you within 3 business days. A short Q&A session will follow the presentation to answer any viewer-submitted questions that you may have. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions anytime throughout the presentation. Joelle, I will now pass it over to you. You may begin your presentation.
Thanks, Amy. Well, thank you everybody for joining. And as Amy already mentioned, my name is Joelle Mosso. I'm Associate CSO for our Produce Division at Eurofins Food. And today we're going to be talking about managing microbial risk with new rapid screening tools. And specifically, we're going to be introducing a new tool that Eurofins has launched called EPRIT, which stands for Enteric Pathogen Risk Indicator. So we'll go into that in a lot more detail on this presentation. For those of you who may not be aware of Eurofins, I'll do a little brief introduction about who we are. So we're a global testing company. We have over 800 different laboratories that are located in over 50 countries around the globe. We have over 200,000 different types of analytical methods that we run. So there's a good chance if there's a test that you need for your business, we probably have a company that is able to do it. Annually, what does that actually culminate to? It adds up to over 400 million tests that we perform for our clients every year. So we're a pretty big company, but what we really do focus on is personalized support for each and every one of our clients. When we look at the United States, which is probably where most of you are all from, we have many different facilities across the country, some of which actually aren't even updated on this slide. Is how fast we grow. But we have microbiology sites that are located all around our United States. We have some pretty unique options with rapid micro express to be able to receive samples in the middle of the night, courier pickups and whatnot. And then pretty much every other type of testing that you may have that would be related to microbiology, we likely have a lab able to support. So if we haven't worked with you, we'd love to. And then, here's all our different sites. The cool thing about Eurofins as well is that if you have a local laboratory that you do work with, you effectively have access to all of our global laboratories through that one lab. So you only have to manage one relationship. That's us in a nutshell. Now on to probably what you came for, which is the agenda today. So we're going to start with just a really basic introduction of microbial risk and how that builds into a testing program. So to provide us a foundation of why we're here and why we started. We'll then take that into really what we're talking about today, which is the risk screening tools and that new tool that we're introducing to the market called EPRIT. So it's always good, I feel like, in a presentation or to connect the dots, I guess, is to lay a foundation of why did we start. And so probably all of you on the line are very familiar with these statistics, but we'll just get everybody on the same page here. So the CDC estimates that 1 in 6 Americans every year get sick from foodborne illness. That's, for people who like math, that's 48 million people every year. Of those, 128,000 are unfortunately hospitalized, and even more significantly, 3,000 people die. So it's obviously not a small burden of illness that we see in the United States, even with our great food safety systems. But the other thing that's not captured here is the economic and societal burden. So we have the economic burden of all those recalls and outbreaks, all the food that gets disposed of, as well as the societal burden of people who can't go to work because they're sick. We see that obviously with coronavirus, but obviously, on ongoing purposes, we have the same issue with the foodborne illnesses and impact to our society. You can see some of the leading causes of foodborne illness on this slide. But ultimately, take-home message here, it's a big problem in the United States, which is why we all want to talk about microbial risk management and food safety. There's many different agents that can cause foodborne illness. These are probably the ones that we talk about most frequently in many industries. So these are some common names you've seen out there: Salmonella, the pathogenic E. coli, so the E. coli 15787, the S-tex, Campylobacter, Listeria monocytogenes has been in the news quite frequently in the last few months, Staphylococcus aureus and the toxin producers. We have some parasites, so Cyclospora. And then if you recall on that previous slide with the foodborne illness impact, norovirus is actually the leading cause of foodborne illness in the United States. So we have our viral issues, not just with COVID, but also in the food industry. Some of these pathogens are pretty easily associated with certain items. So you come to mind, yeah, everyone knows, raw chicken probably has a salmonella problem, but some of them are less so. Regardless, this isn't an exhaustive list, but it does give you an idea between the foodborne illness impact as well as some of these pathogens is what we're looking and talking about today. So given that, most of you are likely in some sort of food safety professional role. And what's really our job as we show up every day? What are we supposed to be focusing on? And we look at all that foodborne illness and see our primary focus is that we should develop and manufacture products that are safe for consumption. That's our main goal that we show up every day to do. We do that by doing analysis and being really reflective about what are the inherent risks associated with the product. So a good example was that salmonella and poultry, right? Or maybe S-tex and lettuce. We see a lot of outbreaks for that. We also have to look at our processes and procedures to see if we introduce risk into the product. That's our other hat that we wear. And we also look at our production environment, and I'm calling out, specifically is that we often think about production environments in regards to environmental monitoring of the facility. But very easily, our production environment could be outdoors. So if we're a grower, growing product in fields, or if we do in kind of processing outside, your production environment is actually outside as well as in the facility. So we also have that lovely job of trying to manage risk in a production environment, some of which we have more control over, like a facility, and some of which we have less control over, like outside. We then take all of that information and all those spinning plates that we keep managing, and we derive a food safety program that also has to meet regulatory requirements. So we have the federal regulations that we have to keep track of, state regulations, obviously, local, and then international if you're exporting product or bringing product in, right? So a lot of different hats that have to be worn by a food safety professional to account for and manage not just production activities, but also all these food safety and regulatory requirements. Today, we're going to focus really on one aspect or one regulatory program, FSMA, so the Food Safety Modernization Act, and their emphasis on risk management. We'll start that just by a brief introduction about the food safety plan required within FSMA from the get-go. So there's 5 components in a food safety plan for a FSMA compliant facility. The first is a hazard analysis, so identify those known or reasonably foreseeable hazards that are going to be happening in your product or your process. You'll then develop preventative controls and write them down and make sure that they are identified to minimize or prevent those hazards. You then need to develop some sort of management of those preventative controls to make sure that they're effective. If you're procuring ingredient and bringing it in, which probably most of you are, there's a supply chain program that you also have to look at for those same types of hazards, as well as mitigations that you may need to apply in your supply chain. Ultimately, hopefully anyone ever has to use it, but you have your recall plan such that you can very expeditiously bring product out from the marketplace and prevent people from getting sick if you ever have a reason to. So today, what's the purpose of really going through this is that the FSMA regulation has actually put a lot of language and emphasis on risk management and really how do we approach our program in a more totalistic way where we're not just looking at things individually. So the overall risk of identification and management and how that may actually adapt over time. So today, 2 of the components that we're actually going to focus on and how that integrates into building a risk management program and using risk screening tools is looking at our hazard analysis. So how can we identify these, microbial, in our case, because we're going to talk about microbial risk for our food process or our environment. And then what are the tools to actually monitor and prove that we're doing that efficiently? So those are the 2 big components of our food safety plan required by FSMA that we're going to talk about today. All right, so kicking off with the hazard analysis and management. The first step that we have to do really is identify those hazards and risks. And so we briefly went over some of those common items, pathogens of concern on our first couple slides, but we look for those and, in a cross-functional team, ideally, look at our supply chains, our ingredients, our processes, and our environments to know what do we need to look for and how do we develop our programs efficiently to monitor them. We then use that information to improve or build upon our good manufacturing practices, our good agricultural practices, or our preventative controls if required, to minimize those risks and really try to reinforce the food safety culture. We talk a lot about and hear a lot about food safety culture these days. It's definitely a priority of FDA and Frank Yanis to really emphasize not just are we crossing all the T's and dotting all the I's and checking the boxes, but are we building a culture that will consistently and ongoing identify conditions that may lead to an unsafe food product. So it's really important. Once we've done all that work, we have to know if we're doing a good job, right? So we have to develop testing programs that first may actually identify the risk. If there's not a history of outbreaks or a known association of the pathogen with the food product, you may have to use testing to identify a risk. You also can use testing to define or support mitigation steps. And certainly you can use them to augment your risk management plan, which is the whole point of everything you're doing in your risk management scheme. It's risk identification management scheme. It gives us this iterative cycle where we conduct hazard analysis over and over. It may change over time, right? We develop control measures in our processes, and then we have to develop these monitoring systems to be able to understand if we're doing well at controlling the hazard that's in our place, and we evaluate those trends. Those trends then go more into the hazard analysis. So it's a constant cycle that may change over time. I think the thing we all need to remember, especially with biological agents like bacteria, is that they constantly are adapting and we need to as well. So our programs should always be a living program in that we evaluate risks and modulate our behavior if those risks start to change. So how do we do that? Well, that's one big component of building an effective risk monitoring program. So you can certainly look, do risk identification to find your risk, but how do you know if your risk over time is changing? How do you know if your program is actually able to assess the information that you're looking for? We're going to talk a lot more about that on the next few slides. But first, just as we started with kind of a baseline of why are we here with the foodborne illness, we'll start with really what are we trying to do with testing. This is a favorite slide of mine, but testing as a tool. So it's really being explicit about what testing is and what testing is not. So let's put it in more of a frame of reference of something that we're more familiar with. Well, many of us probably are more familiar with. So we're looking at this health scale. It's a weight scale. You've probably seen your doctor's offices. And obviously it will tell you your weight. So that's what this test does. The important thing to remember about a test or this scale, right, is that I can get on and off of it thousands of times during the day. But unfortunately, it will not help me lose weight and it won't make me gain weight. It'll just tell me where I'm at. Some scales are more accurate than other scales. So maybe there's a range on the scale where it's 10 pounds. It'll show you in 10-pound increments what your weight is. Well, that's really effective if all I need to know is that I am 10 pounds within wherever I want to be. But if I'm trying to lose 5 pounds, I won't know if I'm successful all that frequently. So you always have to be very familiar with what the limitations of the scale might be. The other thing about scales, right, is that we all know the trick of when do you want to weigh yourself during the day, because your weight changes throughout the day. So if you want to be lighter, you figured out probably when the lightest point of your day is, and it fluctuates otherwise. And you know that over time, you could take your weight many times during the day and have different findings. Overall, in cumulative, you'll have a good indication of what your weight is. Okay, so now we've grounded on what really is a tool by thinking about it in relations to a scale. We'll change it. right? So tests impart information. They alone don't do anything, right? So they're just results that are going to tell you information that's going to be conveyed to you. It doesn't change anything unless you do something about what you saw. So if I go back to my scale example, if I get on the scale and I find that I'm 10 pounds heavier than I want to be, and then I choose to go run, I've now done something about what the information told me that actually changes my reality. And that's the same thing with any kind of test that we're doing in microbiological testing. The testing alone is just going to convey information. It doesn't change it. The other thing with testing is that there's limitations, right? So it can have a different range. It can even be broken. And we all had the probably horror of a broken scale when we go to the doctor's office and we think we're great because at home we're great, but our scale is actually broken at home. So that is true with any kind of testing, inclusive of microbiological testing, is that we need to be very cognizant of what the test limitations are and realize that they can be still performed, incorrectly, or they could be broken. So tests are just information. We shouldn't just, blindly accept whatever comes out of, an instrument, and we need to think about it critically. Just like the scale, tests are just moments in time. You're going to have fluctuations of results. So a data point isn't necessarily all that useful. A cumulative data set is very, very helpful generally. So knowing that, the biggest one we need to remember is that testing is just a measurement. It's not a mitigation. So as we think about maybe our testing program in a different way from, potentially like, well, I'm trying to control salmonella, to I'm trying to control the risk of salmonella. We recognize that testing is just a measurement. It's not a mitigation. So if we want to change our program, we can do so. It's not going to, impact the true food safety of our product. It's what we do with that information that does. So it's to tell us something, not to control something. And effective testing, a really well-designed program, allows us to make better and better decisions. So that's really the biggest things when we start to set out to create our environmental, or our risk management program, is that we really want to be explicit about what we're trying to do with our testing and what it's going to be telling us. So what's the purpose? We need to be very deliberate about your testing purpose. Is it surveillance? Is it risk assessment? Are you trying to actually first ascertain what is the risk? Is it a verification program to see that your preventative controls are working, or some other process control that you've put in place? Perhaps you're actually in an investigation. Maybe you had an event and you're trying to get to root cause. All of these things may be places where you input testing. Often we think, well, my risk is salmonella or my risk is STEC. So we think we must go out and look for the pathogen, itself, and that's maybe what we think our testing program should be. Or we might be doing monitoring of conditions that are favorable for the pathogen. So maybe we're looking at Listeria species in our environmental monitoring program. We're not necessarily looking for the pathogen, which is monocytogenes. That would be on that list of illnesses that we get to, 48 million per year. But we're looking for conditions that would identify where the risk of monocytogenes is high. So if we're starting to parse out, right, the difference between the risk of the pathogen explicitly and the risk of conditions which may be favorable for the pathogen. Another one that I listed was EB, so Enterobacteriaceae, which we'll go into a little bit more in the next few slides. But that's often used as a group of organisms that's indicative of salmonella. I keep driving home this point, but testing doesn't make something safe. So a non-detect test, or a negative that you may get, especially if you're doing pathogen testing, can be of limited value. It certainly is telling you something. You didn't find anything. But if you're actually trying to find a way to minimize risk or ascertain the total risk that you may have in your food system, negative tests can actually be of limited value. You have to really think about what you're trying to do and what the purpose of your testing program is. That's focusing on that value versus activity. So just to be a little more explicit about that, value in testing is how good is the information that you're getting to be able to dictate what your next action needs to be. So a negative doesn't really impart anything to you to go do anything differently. it's kind of like getting on a scale and finding out you're exactly the weight that you need to be. But if you find out you're the weight that you don't want to be, you now have incentive or motivation to go do something differently. So when we evaluate our programs, and especially in regards to managing risk, is that we really want to be getting feedback that allows us to change our behaviors and help minimize or drive risk down. So we want to focus on value versus just activity. I don't care if you do thousands and thousands of tests, swabs, potentially, or product testing every day, if it doesn't help us change the risk. So we really want to focus on value versus just activity. Same thing, different words, find value in your results. And sometimes that may mean you need a different approach. And there's nothing wrong with actually changing approach when you're trying to design a more informative way of getting to the next step in your process. So how do we find the value in testing? We start with what to test for, right? So direct pathogen tests are certainly an option. Indicator organism. We may even have indicators of biological materials. If you think about that, it's like ATP swabs. They're not necessarily indicative of bacteria, but they certainly can give you a good indication if something is clean or not. Is your method fit for purpose, right? So if that 10-pound range scale it isn't all that effective when you're looking for a 5-pound weight loss, are you sure you're using the right method for what your purpose is? Those in microbiological testing include sensitivity and specificity. So basically, is your negative truly negative? Is your positive truly positive? Do you have a reliable and repeatable testing that you do so that you can trust the results that you're going to get every day? The time to your result, obviously we all live in fast-paced worlds, and if you've got a long shelf life product, you probably have no problem waiting days to get your results. But if you're in a fresh perishable product, minutes matter. So that's a very appropriate and important characteristic for fit for purpose. What's the output of the test? So what information are you going to get from it? And does it meet your expectations? And sometimes those expectations may actually be your customer's expectations. And most certainly, price is always of importance. And how does that fit into your total plan? Remember, testing is just a tool. So it's one component of everything that you're going to do to actually minimize risk. So it all has to fit within your budget. Let's talk about direct pathogen testing. So certainly we can look for pathogens directly to monitor risk. So if we know Salmonella or STEC is our problem, we can definitely target looking for those directly. The issue with a lot of pathogen testing is that they're often at very, very low prevalence, which is good, right? We don't want tons and tons of pathogens out in the food environment. But it makes it more challenging when it comes to testing to actually find something. So you could do many, many, many different tests, thousands even, and maybe not find the target. And it doesn't necessarily mean it's not there. It's subject to lots of statistical sampling requirements and just the likelihood that you'll find the cell that you're looking for on any given day. It's kind of that needle in the haystack concept, is that it's pretty difficult to find a needle in a haystack, but it doesn't mean it wasn't there. And that's the challenge with pathogens. And it's very difficult if your test can't, quote unquote, see what you're looking for. There are also quantitative methods for pathogen testing, and those are based on culturing. You see a plate here of XLD, which is a salmonella type media, and then you see salmonella growing. So that's based on culturing and the biochemical appearances of these organisms. The problem with these tests, they're great, but they can be very slow. It take days to get to results. Again, not the most expeditious thing if you've got a short shelf life product or need to make decisions quickly. We do have rapid methods that are available to us, and we'll talk about some of them in the next few slides. They definitely offer faster testing, but they can still be limited due to the low prevalence. We still have the needle in the haystack process, or problem rather, where we just don't know if our sample is going to pick up a low prevalence of a pathogen. And then we think about, the regulatory impact of testing for pathogens. There's a lot of potential implications if we find a pathogen, which can make it difficult in the food processing environment, especially if you're getting into risk assessment or investigation to try to understand better what's going on. Because if you end up testing and find out too much information too soon, you could potentially find yourself into a recall situation. So these are all considerations when we're setting up our testing program for risk management is that we definitely can do direct pathogen testing, but there's some things we need to think about when we do that. As an alternative, there's indicator tests that we can actually use. So indicators provide insight to an overall food safety or in some cases quality of a sample, product, or process. They can highlight the potential for risk. So we're not actually looking for the agent itself that causes the problem, but we're looking for groups of organisms or indications, hence the name, that may be a risk that's present. They're not conclusive that a pathogen is there. So if you think about Listeria species, we're identifying conditions where Listeria species can grow, which could mean monocytogenes could be there. That's an indicator for monocytogenes. Some indicators are very, very tightly correlated to a pathogen. Listeria species is a good example. That's a smaller subset, just the other Listeria species that are in the same species. But others are less so. And so you have to really evaluate the value of the indicator and the quality of the data you get. Common indicator tests that are used in the food industry are used for trends like sanitation control. We'll go into some of them, but for the sake of this conversation, Enterobacteriaceae, E. coli, coliform, these are things that we talk and hear a lot about. They're very helpful in assessing quality and pathogen risks of ingredients in some cases, and we see a lot of it with indicator testing with water quality and our sanitation processes. So we see a lot of indicator use in the industry. It started out many, many years ago, and we still just continue to see that trend going forward. Indicator tests are often quantitative tests. So they provide information on the quantity of the organisms in a sample. But that, if we think back on that red plate with the black colonies that were growing on it for Salmonella, is that it generally takes longer to grow out and culture those organisms in a quantitative method than it does if you're looking at a more rapid PCR or genomic-based method, which we'll go over in a couple of slides. Indicators can be, and I'll emphasize can be, less informative of pathogen risk in certain situations. So, you may have a scenario like take leafy greens, E. coli, coliform, and Enterobacteriaceae are normal microflora on the leaves of lettuce. So they wouldn't necessarily be a great indicator of a pathogen because you would expect them to be there. So indicators can be really useful in some scenarios and less so in other ones. So it's that normal microflora problem and that they are actually broad groups of organisms. So what are those broad groups? So just, here's ones we talk about often. Enterobacteriaceae, generic E. coli, coliform, fecal coliform, Listeria species, and you can see on this figure here, there's groups within groups. So Enterobacteriaceae is inclusive of total coliforms and E. coli, and also the pathogens. So it's a much bigger group than if I just looked at fecal coliforms or E. coli itself, if I'm looking for 0157. So finding the right type of indicator is really important, and understanding what those organisms are and what they represent is also very important when you're picking an indicator as a test. So I've mentioned a few times now quantitative versus PCR. So just very briefly, so all these could be presentations in and of themselves, right? We have quantitative methods that I've really emphasized is about growing them on plates or getting to a number where we know something's there. So they're designed to give us information about the number of the organisms present. They are based on biochemical properties to help differentiate the organisms. In most cases, not all, but in most cases, they are slower to results than many of our genomic methods, which we'll talk about one here. There's many types of genomic methods, but the one relevant today is PCR. So polymerase chain reaction, it's the DNA of your cells. So just like if you were doing CSI and trying to find somebody at a crime scene, we have that same type of technology for our foodborne pathogens. Most PCR methods are generally qualitative, so they are yes, no. They're not going to tell you how many cells are there. They are designed to amplify a certain gene target in the DNA for a pathogen, or anything that you're analyzing. So it's a specific target that you're looking for, and it's still dependent on growth. So PCR, it generally follows an enrichment period to get the number of cells to a level where we can actually detect them with PCR. Albeit PCR could be done without enrichment if you have a high concentration of something that you're looking for from the beginning. Positives, or presumptives, that are found by PCR reactions means that DNA is present. So effectively, something was there. It's the CSI crime scene. We knew you were there. What we don't know is if you're living. So that's what we learned with PCR, is that DNA is present. Whatever our target was there in our sample. So here's our new tool, the EPRIT introduction. So it's the enteric pathogen risk indicator. It is a qualitative method, which means the presence or absence. It is a screening tool, and note the asterisk, we'll go into that in the next slide. But it screens for Salmonella and Shiga toxin producing E. coli, so our STECs, and that would be inclusive of 0157. It is not conclusive of a pathogen's presence, so it is a screening tool. It's a rapid PCR method, so if we think back on that last slide, quantitative versus PCR, it's faster than if we tried to grow out these organisms in a culture or quantitative method basis. It's based off of DNA for the pathogens of interest, but it also includes other gene targets for PCR that are not conclusive with pathogens. And effectively, it's a new rapid diagnostic tool for food companies' toolkits. So what's the asterisk mean about screening tests? So screening tests are used to give insights. They're not necessarily giving you conclusions. Common ones that we have, that we experience in our day-to-day lives are heart rate and blood pressure tests, blood panel tests. And effectively, if you go in and you have a high blood pressure reading, what that is a signal to your doctor to say there's something else may be going on. So a risk may be present, but we need to do further testing to really understand what's going on. And that's how we want to think about risk screening tools in regards to this presentation and the EPRIT tool, is that it is a way to give us insight about risk. It is not conclusive of risk. So risk screening does not only detect pathogens. It has, however, been validated to prove that it will detect Salmonella and STEC if they're present in the sample. We have completed that validation with our colleagues in Europe, Eurofins Gene Scan, to prove the inclusivity of 120 different Salmonella enteric strains, 50 STEC strains, including 0157 and our top 7 STECs. They have various enrichment times that are rapid, so 10 to 24 hours, depending on what you're testing and what your scenarios are. EPRIT is a real-time PCR platform. So what that means is that it's monitoring, this instrument is monitoring data throughout the PCR process. And effectively what you're doing is taking maybe that one gene copy that you have in your sample and you're amplifying it exponentially over time. The instrument is able to read that growth and determine if your sample is positive or negative. The nice thing about real-time PCR, as opposed to older methods developed in the 1980s and whatnot, or the gel-based methods, is that the instrument itself will be calling the positive or negative as opposed to a human. And there's a lot of more consistency in each reaction, and it also provides consistency, therefore, between labs and runs without any human interpretation. EPRIT detects genes from these enteric pathogens that we're talking about, Salmonella and STEC, but it will also detect other non-pathogenic organisms. And no data that is generated by the real-time instrument PCR or any part of the process is actually able to differentiate signals from pathogens or non-pathogens. So it's truly just an indicator of risk. It is a screening tool. So what does an EPRIT risk not detected mean? It means no gene targets were found in that sample, which effectively means that sample does not include STEC or Salmonella. If you find an EPRIT risk that is detected, what that may indicate is that a pathogen may be present. Gene targets are not conclusive of Salmonella or STEC. The assay will detect more than just STEC and Salmonella. That's the design of the test, because it is a true screening test. Its result alone is only a risk indicator. So what it is effectively telling you is you have conditions and presence of organisms that may indicate a risk. It definitely doesn't conclude that a risk is present. So if you're looking at like the process flow for EPRI, this is like a good flow. So you've got your initial sample, you're going to do an initial screening for risk of these organisms. You as the client have the option to stop here with the indication of risk. You do not have to proceed to know if the pathogens are actually there. All of the EPRI validations have been built off of AOAC and GeneScan back gene validations. So methods that enrichment conditions and whatnot that actually give you more faith in the culturing in the enrichment phase. If you find a risk and want to know, okay, we've been told an EPRI risk is present. What does that mean? Can I know what is there? Yes, you can. And the unique thing about EPRI is that it allows you to run that exact same lysate, which is the part of the sample that goes onto that PCR instrument, to be run for the same pathogen of interest. So it minimizes sampling error and having to resubmit samples, and allows us to test that exact same sample within a few hours to figure out, okay, is it Salmonella? Is it one of the top 7 organisms? Another option after that is that you can choose to go to cultural confirmation. So why, for those who maybe are not as aware of with PCR methods, is that still the gold standard is to know whether or not the organism is living, because DNA can be detected on PCR even if the cell may be non-living. So cultural confirmation lets us know that yes, the signal was there and the organism is living. The EPRI method allows for that as well. So it gives you a lot of different flexibility and options of where and when you want to stop on the process. So the EPRI method has been built off of the existing AOAC vaccine kit method. What that means is you have the ability to control how much you know about a risk and when. It can detect the risk of Salmonella and STEC in one reaction. I'll emphasize risk, right? Not necessarily the organisms themselves. EPRI has some additional tools that are also consistent with some of our other vaccine platforms, but gives you more confidence in the results. So we have what's called pre-eraser. It's a vaccine treatment that actually removes free DNA from the sample. So what is free DNA? Well, free DNA is basically DNA that may be in your sample that you submit, in your swab or your product, but that DNA is not found within a bound living cell, right? So it doesn't have a cell membrane around it. Effectively, the pre-eraser is an enzyme that can chew up and degrade free DNA that may be in your sample. So the DNA that is bound within a living cell will not be impacted, but DNA that may be present because of dead cells can be minimized with the DNA treatment called pre-eraser. It can be a very effective tool to help minimize what I would call noise to the industry, where I don't care if the cell isn't living, I'm only worried about living organisms that can cause illness. So this is a way that you can try to get at that with this nice treatment. Another thing that EPRI has is a UNG technology that comes with every EPRI reaction and kit. What that is not to get too into the weeds of it, but effectively there's nucleotides, which are the building blocks of DNA and build up those genes. We've replaced one of those building blocks with uracil, which is normally found in RNA and not in DNA. And because of that, we can actually use an enzyme at the very beginning of our PCR run that will degrade any prior PCR product. So we talked about how PCR amplifies a target that may be present in your gene over and over and over, so you have millions of copies. Well, if those copies were to get into somebody else's sample or into a next PCR run, that potentially could give you a false positive. So it would be a lab error. This actually degrades that as being a possibility because it has replaced uracil with the thymine in our PCR reaction, and that enzyme will chew up any of that product before we start amplification of a new run. It minimizes the concerns of lab PCR contamination. So between the two tools, the pre-eraser, the DNase treatment that can help minimize detections of non-living cells, and then this one, is that it also gives you confidence that your lab isn't making any errors in detecting what could be prior PCR amplicon detections. So importantly, we've introduced a tool, but really how do we use it, right? That was the whole point of, testing is a measurement. It's not a mitigation. So how do we use it and make it an effective tool in our food safety plan? We can use it to verify the efficacy of our good manufacturing practices, our good agricultural practices, and certainly our preventative controls. We can use it as a screening tool to evaluate and explore risks that may come from certain items or certain suppliers. So if we think about, is grower A or grower B providing product that is safer or riskier than another, person, we can use it to explore risks in that way. We can also look at using EPRI as an environmental risk tool, so sites or water sources and input. It could be used in the validation of new procedures or mitigations. Perhaps you're changing your sampling plan and need to show equivalency, or you're looking at a new treatment that is going to reduce concerns of pathogens or something of the like. Those are options where you could do it. Certainly sanitation practices. I mean, most people, their environmental monitoring programs are probably the biggest surveillance programs that they have in place. And if we think about that, EPRI is a great tool to be able to facilitate even larger risk screening and assessment. It is a broader capture of risk than just direct pathogens, so it can help minimize some of the needle in the haystack issues. So it gives you a little bit of broader screening to know if there's a risk. It is a rapid PCR method, so it is faster and more focused than many common indicator tests. ATP is obviously the fastest. That's like, in a few minutes, but it's not necessarily reflective of bacteria, let alone a pathogen. Enterobacteriaceae and E. coli coliforms are great indicators in many scenarios, but if they're common microflora, they tell you very little about a pathogen's correlation or presence. And this initial screen with EPRI can be paired with an AOAC validated method if found. So if we think back to that process flow, when you find an EPRI risk is present, you have the option to test that exact same lysate using the validated AOAC kits and actually have your results reported as a validated method, because how this was designed. The test was designed to use all the same conditions of our validated AOAC methods, albeit you're just getting information sooner in a broader sense to even identify first if the risk is present. So it does give you an option to get back to a validated method if you do find an EPRI risk in your sample. Importantly, it can be paired with other indicator testing. So if we think about common environmental monitoring, we do a lot of Listeria species testing as an indicator of Listeria monocytogenes. We have yet to have a screening tool for Salmonella or pathogenic E. coli, and EPRI can certainly be that. It can also be paired with Enterobacteriaceae or generic E. coli or coliform. And effectively what it does is it is tighter in that it is looking at more of a pathogen risk than some of those broader groups. So it can actually help give added depth to understanding those broader indicator groups if you're truly trying to manage the risk of these pathogens. Facilities that have high risk for Salmonella and pathogenic E. coli have been out of luck if they're just trying to use Listeria species. Those are different organisms, behave in very different ways. This tool gives you as an option a way to screen for risk for enteric pathogens that are Salmonella pest. So if you think about facilities that might be dry facilities or facilities that have products that are highly associated with these pathogens, perhaps nuts or lettuce, those sorts of things, that it adds to the toolkit to get a better idea of how your environment may be changing over time. All in all, EPRI has a lot of advantages in that it is a risk screening test. So it's controlling the information, it's rapid, and it offers the ability to do a risk assessment with a closer correlation to pathogens than some of these broader groups that you may have used historically. It covers Salmonella and S-Tech in one reaction, so we think back to that fit for purpose. Price is a big driver in how we're designing our different programs, and EPRI can help with giving you more opportunity to do more value in your testing than just looking at activity. Importantly, and probably very significantly, is that it allows you a pathway back in a very rapid way to an AOC validated method within hours. So you don't have to submit new samples and be subject to sampling error, as well as you have the option to know for sure is a pathogen present or not if I find a risk. That gets us back to, the very beginning when we started talking about building an effective monitoring program. It's like, really, what are you trying to do with your testing? Testing is just a tool that allows you to verify activities, identify risks, or allows you to improve your food safety program. So it's really, really important that we select the right tool for the activity that we're trying to assess and really start to parse apart, like Salmonella or STEC may be what I'm worried about, but how do I best assess the risk from those organisms? And it may be a different approach than just direct pathogen testing. Importantly, and irrespective of what test or tool you choose, you really need to use your data to drive for better programs and reduce the risk in your supply chain. That's the whole purpose of what we're trying to do with testing. If we have millions and millions of data points of never finding something that actually informs us to change our behaviors, we'll never start to drive risk down. So we really want to find tools that allow us to better identify risk, to better change our behaviors, to minimize those foodborne illness events that happen every year. You also need to remember risk is dynamic. Things change all the time in your food processing plants and in your supply chain. So always keep that iterative cycle of looking at risk and adapting to risk and trying to see, are we being effective at controlling it? And picking the right tool is really critical to that. So you want to be able to identify when trends start to change. All of that basically is just saying, design your testing program with a fit for purpose. Think about all the characteristics and information that you need before you start testing, and then design the best tool and approach to do that. Eurofins is here to help as you try to think about building that system. So we pride ourselves in being partners for your testing and risk management needs. I often say is, testing is what we sell, but service is really what we provide. And ultimately, it's our job to make your job easier. We also provide complementary data analysis tools that can help with your tracking and trending. That starts with online ordering and actually follows your samples throughout our laboratory. And as results populate, you'll see them in real time on the system. It can allow you to visualize better the risks that you have at various times of the year. This is especially true if you're using floor mapping tools that we have. So if you're thinking about environmental monitoring or even grower or site location issues, you can actually show where you're taking your samples and see over time how your trends and where your positives might be. So you can start to hone in not just on how many samples have we done and what were the results, but what trends are we seeing and how can we better develop our system to minimize our risk. Design your total testing program with that fit for purpose and recognize you can always change. The point is to find value and not to just drive up activity of what you're doing with your testing. Make your testing dollars work more for you. So be adaptive in your approach and be proactive to try to find ways to minimize risk. Our purpose, right? If we think back to where we started in the very beginning of this slide, is our purpose to show up every day is to reduce risk and reduce those events of foodborne illness in the United States or whomever we may be providing product to. So it's our job to control the food safety of our products and really think about risk screening and risk management in a way that is a continuous improvement project. And with that, I'll say thank you, and I will open it up to questions.
Thanks so much, Joelle, for your presentation. And just while we're getting started with the Q&A here, I would like to add this question to the screen here again. If you listen to the presentation today and you would like someone from Eurofins to follow up with you, go ahead and select yes there. And there have been just a few questions that came in regarding the presentation itself. Just to remind everyone, you will get a copy of the presentation as well as the recording within 3 business days. So expect to see that. And then there's some questions that have come in here. So Joelle, the first question, how does EPRI work with Listeria?
Right, so EPRI is looking at gram-negative enteric pathogens. So in broad strokes, we're looking for Salmonella and Shiga. Listeria, we mentioned it many times in the presentation, but it's a gram-positive and it has very different characteristics from those enteric pathogens. So historically, we've used Listeria species as an indicator of risk for Listeria monocytogenes. And most certainly, many of our food plants and facilities and our food products have risk of Listeria monocytogenes. So that's a great indicator. Where we've struggled, I guess, over time is that just because you do Listeria species monitoring in your facility does not mean you do not have Salmonella risk and E. coli risk, pathogenic E. coli risk. But we have not had as an indicator for those organisms. So the nice thing about EPRI is that it can be paired with Listeria species testing. They have different characteristics, which means we need to grow them differently and detect them differently. But in cumulative, that bundle between EPRI and Listeria species gives you a much broader snapshot of the risks that your facilities may have. So if you think about environmental monitoring programs, in this case, could be product too, but environmental monitoring programs is that we'll often look at doing some quantitative tests in combination with our Listeria species tests. So we might do Enterobacteriaceae or coliform or E. coli, generic E. coli. In the same time, to get a better idea, is our cleaning processes and sanitation processes, good? Well, we get counts on Enterobacteriaceae or E. coli coliform. We really don't know if there's a risk of the pathogen per se. All we know is that those organisms were there, maybe our cleaning wasn't sufficient. EPRI allows a much tighter correlation, much similar to Listeria species, that gives an indication of the pathogens themselves. So it's a nice bundle if you put EPRI and Listeria species together, because it gives you a more comprehensive picture. And it's probably worth noting, right? We talk a lot about, and FSMA talks a lot about environmental monitoring, but it is not limited to Listeria. And if you have risk of the Salmonella and Shiga, either from your product or just from the environment, you should be screening for those as well.
Great, thank you. The next question that came in, is there a limit to the product type that can be tested with EPRI? For example, would it work with alcoholic products?
Interesting.
I know.
Yeah. There's not necessarily a limit. As with any type of test, it needs to be proven to be the right tool, right? So that would be something we'd have to evaluate and show that the method is verified for that product. I would imagine alcohol might be a challenging organism, a challenging matrix to do any kind of PCR on, as well as to prove that your organism can actually grow in it, because if it's high enough in alcohol, it's probably actually a kill step for most of these organisms, but it's something we could look at. And that's an important point too, is that, with any test, it's understanding those limitations and recognizing that you can find a test that may give you the value of the information you want, but you still need to verify that it will work in the matrix that you're trying to test, in this case, an alcohol product. So that's something we would want you to reach out to us on, and we could talk about whether or not it'd be appropriate or not.
Wonderful, thank you. And it looks like we have one more question here. I'm just catching up on them. If an EPRI detection is found, would you have to continue to the pathogen test?
No, that's one of the nice things about the EPRI test, is that it allows you the control of where you stop. So again, another example would be looking at, Listeria species is probably a good example, is that a lot of people find, okay, there's a Listeria species present, I'm going to just assume the risk is there of monocytogenes, and I'm going to go do cleaning and sanitation. And so I'm just going to act on that information. You don't have to proceed to the next step. Now, that being said, you may want to know, okay, there's definitely a risk of this pathogen being there. I might want to know, is that risk there? Is it finished product? Is it something that I may need to react to? Maybe it's a food contact surface. At which point then you probably would want to know if it's present or not, and not just stop at the risk level. The real value of EPRI, besides that it is a risk screening, so it's a little broader, is that it allows you that flexibility to design your program in a way that makes the most sense for you. So it's really driving at that value versus activity that I kept reiterating over the presentation. what's the value to me? The value to me is knowing the risk is there and I'll do something about it and move on, or do I need to know if the pathogen is there? And the nice thing about EPRI is that it's been developed such that it gives you that pathway back to a validated AOAC method for the direct pathogens. So if you do get a hit, you have a way to follow up and know whether or not the pathogen is there using an AOAC validated method.
Wonderful. Thank you so much. And it looks like we are caught up. In case we have missed any of those questions, we'll certainly follow up with you after the presentation. But that wraps up the Q&A session. I want to remind everyone, as I mentioned a little bit earlier, a recording of the webinar along with a copy of the slides will be sent to you within about 3 business days. Thank you again, Joelle, for your presentation today. And thanks everyone for attending.
Thank you, everybody. Thank you, Amy.
Thank you.
Bye.
Bye.